Once again, welcome back. In this part of video, I'm going to talk about neuroglia or glial cells. As I mentioned before, that neuroglia or glial cells are supportive cells of the nervous system. They are not functional cells like a neuron. Okay, so let me talk about different types of neuroglia. One type of neuroglia is called astrocytes. Astrocytes means that they looks star-like this. Okay, for example, this is a cell, and this cell has different projections. Okay, and they looks like a star. Okay, star-like. So that's why they are called astrocytes. So let me talk about the different parts, the different functions of the astrocytes. It's so important. Okay, first function of astrocyte is that it has role in blood-brain barrier. Bladder brain barrier. What's blood-brain barrier? Look here. For example, this a blood vessel. This a blood vessel which passes through the central nervous system. So there is a communication between this blood and this nervous tissue is not freely. Okay, they are not free. Something that passes with this endothelial cells or this blood layers to go to the nervous tissue, so they can't pass this area easily because here is a barrier. Barrier means this area is tighter, so this barrier is between this blood and this nervous tissue. That's why this is called blood brain barrier. Okay, let me talk about the components of the blood brain barrier. Blood brain barrier is made of, firstly, in a bladder that passes through the central nervous system, we have endothelial cells. So first part of the blood-brain barrier is endothelium or endothelial cells. Okay, this is the first part. Second part of the blood-brain barrier is between these endothelial cells. There are proteins. Okay, and this collection of proteins are called tight junction. They are called tight junction. Okay, that's two. And these endothelial cells are not fenestrated capillary or fenestrated endothelial cells. Anyway, the third component of this blood-brain barrier is this endothelial cells basement membrane, or this is called basal lamina, because you know basal lamina is a part of the basement membrane. So this is called basal lamina. Okay, basal lamina. The fourth part, look here, this is endothelial cell. These are tight junctions. Around these endothelial cell we have this transverse section like this. This is a marker. If I cut it transverse and then I show it like this. Okay, so this is the basement membrane. After this we have some cells that these cells are called pericytes. These cells are called pericytes. Pericytes, and these pericytes are always around the blood vessels. Okay, after this pericytes, you know, this is an astrocyte. This astrocyte has these parts, and these parts are called end feet. These are called end feet. So this end feet of the astrocytes cover this capillary, and they acts as a barrier. So that's why we say that astrocytes has role in blood-brain barrier. What are the components of the blood-brain barrier? Endothelial cells, tight junctions, basal lamina, astrocytes, and pericytes. Okay, so this was about the structure of the blood-brain barrier. Okay, here this is endothelial cell, these are basement membrane, these are tight junctions, this is pericytes, and these are end feet of astrocytes. Now in our brain we have some areas that they do not have this blood-brain barrier, their lack of blood-brain barrier, and as a collectively these organs, these areas are called circumventricular organs. Circumventricular circumventricular organ. Circumventricular organ. Why they are called circumventricular organ? Because in our brain we have some cavities that they're filled with CSF. For example, we have two lateral cavities. One, this called two lateral cavities. This called third ventricle, or these are called two lateral ventricle, and this called third ventricle, this called fourth ventricle. So these organs, these area around this third and fourth ventricle, and that's why this called circumventricular organ. Circum means around, and ventricular means around these ventricle, which are CSF filled cavities. Okay, let me talk about these organs. Some of these organs are sensory, the sense our bladder composition, sensory. Some of them are secretory. They secrete some things, some hormones within the bladder. Some of them are secretory. Okay, so now let me talk about them. Okay, here is an area. This is third ventricle. This is third ventricle. Here is a little bit large. Okay, this is third ventricle. From here there is an area between the fourth ventricle and third ventricle, and this CSF cerebrospinal fluid passes through this area. This is called cerebral aqueduct. This is called cerebral aqueduct. So this is fourth ventricle. This one is fourth ventricle. We will talk on these ventricles in future lectures. This is called fourth ventricle. This is the roof of a fourth ventricle. This is the floor of fourth ventricle. You know, this is midbrain, this is pons, this is medulla, this is cerebellum. Okay, this anterior, this is posterior. So in this floor of the fourth ventricle, here is the area, the end of this fourth ventricle, which is called obex. It's also called obex. So here in this floor of the fourth ventricle, here we have some neurons. That collection of these neurons are called area postrema. Area postrema. Let me write it here. Area postrema. Area postrema. They are collective or collections of neurons that has capability of sensing CSF and also blood. In our blood, if we have toxins, if we have some drugs, if we have some things, so it will sense. Okay, and in this area postrema, for example, this is area postrema. In this area postrema we have some neurons that are called chemo trigger zone. Activation of this chemo trigger zone causes vomiting. It stimulates vomiting center. For example, it stimulates nucleus of tractus solitarius, which has role in autonomic control of different organs in our body. Anyway, this chemo trigger zone causes of vomiting. So in a nutshell, I can say that area postrema is a sensory area, a sensory circumventricular organ, which has role in sensing of chemical substances, toxins in our blood plus in our CSF, and also it has role in regulation of our blood and CSF hemostasis. Okay, and when this sense toxins, it causes activation of vomiting reflex and causes vomiting. That we will talk on vomiting in future lecture, inshallah. Okay, so this was area postrema. So one sensory area is area postrema. Okay, let me talk about another one. This is third ventricle. This is third ventricle. This is posterior wall of the third ventricle. This is roof of the ventricle. This is floor of the ventricle, and this is anterior wall of the ventricle. Okay, at the roof of the third ventricle, here we have a bundle of axons which are called tractor, and they connect some areas together. They connects, for example, some area between the hypothalamus and hippocampus anyway, and they has role in our limbic system. So this tract is called fornix. This is called fornix anyway, fornix. Okay, under the fornix, this is fornix. Under or beneath the fornix, we have an area. And this area is called subfornical organ. This is called subfornical organ. This is called subfornical organ, subfornical organ, okay. Subfornical organ is also sensory. So one sensory organ was area postrema. Another one is subfornical organ. Subfornical organs sense two important things. It's sensible to angiotensin 2. For example, when my BP or blood pressure goes down, and this time my system will secrete angiotensin 2, and this angiotensin 2 has a lot of function. One function of angiotensin 2 is that it will stimulate this subfornical organ, okay. And after this subfornical organ will stimulate that. I'll talk on this later. It will stimulate the hypothalamus, and here is a nucleus which is called supraoptic nucleus, and it will stimulate this nucleus, and this nucleus then causes secretion of ADH or antidiuretic hormone, which causes to increase my BP, okay. So this is sensible to angiotensin 2. Another one, this is sensible to our blood osmolarity. And when our blood pressure or BP goes down, so our blood osmolarity goes up. And I have talked about osmolarity, about all these things in previous lectures, okay. So it's sensible to two things, to angiotensin 2 and also to blood osmolarity, and both of them triggers ADH release, and we will talk about ADH in endocrinology, and then it causes to increase our BP. So this was the second circumventricular organ, okay. Our third is circumventricular organ. That this third circumventricular organ is also sensory. It's also sensory. Where is the location of this? Here, this anterior wall. Here we have mammillary body. Here we have anterior commissure. Here we have anterior commissure. Between these two areas, this area is called lamina terminalis. This red area is called lamina terminalis, okay. So in this lamina terminalis, here we have an area that it's lack of a blood-brain barrier, and this area is called organ vasculism of lamina terminalis, organum vasculism of lamina terminalis, and it's also sensible to our blood angiotensin, to our blood osmolarity, and also it causes to increase our BP, and also in some evidences they say that it secretes some proteins and some other things anyway. So these were sensory area postrema, subfornical organ, organum vasculism of lamina terminalis, okay. Now let me talk about the secretory part of circumventricular organs. You know, we have here a gland which is called pituitary gland, okay. Pituitary gland, we have two parts of pituitary gland: anterior pituitary gland and posterior pituitary gland, okay. Let me drink a little bit water, okay. Let me talk about secretory organs. This pituitary gland, this anterior part, and this is posterior pituitary gland. You know, posterior pituitary gland is part of our central nervous system, and anterior pituitary gland is not part of our anterior pituitary, not part of our central nervous system. We will talk on this in the future. Now, anyway, here we have in this pituitary gland, in two areas we don't have a blood-brain barrier in pituitary gland. In two areas, where this area, for example, this pituitary gland, this area is called this hypothalamus, okay. And this area of hypothalamus is called median eminence. It's continuation of median eminence. It's a protrusion, okay. Here in median eminence, there are some neurons that these secretes different types of hormones within the blood, okay. Then these hormones go to anterior pituitary gland, and then from the anterior pituitary gland these come out once again and causes it stimulates different cells or glands, and after them these secretes other things. For example, this secretes LH, this secretes growth hormone, this secretes TSH, and so on. So the in pituitary gland we will talk that here is a circulation which is called hypothalamo hypophyseal portal circulation, and we will talk what's portal circulation, what are these in pituitary gland. So keep it in your mind that we have one area that's in median eminence that they do not have blood-brain barrier. And what's its function? These neurons secretes many hormones here. For example, growth hormone, releasing hormone. For example, let me give you an example. Here is a nucleus, okay? Here is a nucleus in median eminence. This nucleus is called arcuate nucleus, okay? So this arcuate nucleus releases a substance, a hormone which is called growth hormone, releasing hormone, growth hormone, releasing hormone, growth hormone, releasing hormone. It means that this hormone will come here in anterior pituitary gland, and this hormone then stimulates a group of cells. Okay, here is a group of cells in anterior pituitary gland, and this growth hormone releasing hormone, when stimulates this group of cells, this group of cells then secretes within the blood growth hormone, so if we have blood-brain barrier here, so these molecules, these growth hormone releasing hormone, these are proteins, they can't enter the blood vessels. So if they can't enter to blood vessels, they can't release growth hormone from anterior pituitary gland. That's why we don't have blood-brain barrier in median eminence. That's one area. So this is secretory part of the circumventricular organ. Another part here in posterior pituitary gland, which is the part of the central nervous system. Here we have two nucleus. One is called paraventricular nucleus, and another one is called supra-optic nucleus. For example, this supra-optic nucleus. When it is stimulated due to presence of angiotensin 2 or high osmolarity of the blood, this supraoptic nucleus releases ADH, and ADH is a hormone, a protein. It's a peptide, so it cannot enter the blood vessel. For example, there's the blood vessels of anterior pituitary gland, so that's why when it releases this ADH, it will enter to the blood vessel. That's why we don't have a blood-brain barrier in posterior pituitary gland, okay? And also, for example, it's paraventricular organ during suckling of milk by the baby, or during pregnancy, during labor or baby labor. It causes to stimulate paraventricular nucleus, and paraventricular nucleus releases oxytocin. It releases oxytocin is, we will talk on this in future lecture, that oxytocin is also called love hormone. It has role in love, and also it's a cuddle hormone anyway. So this oxytocin will enter the blood vessel during the labor and during the suckling and during the love emotions, okay? Anyway, so this is the fifth area that's also secretory, that do not have blood-brain barrier. We studied five area postrema, subfornical organ, organum vasculosum of lamina terminalis, and then median eminence. Then we talked about the posterior pituitary gland. And let me talk a little bit about another area. Here is another area in our brain. This area is at the back of this third ventricle, which is called pineal gland. Here is a gland which is called pineal gland, and pineal gland releases melatonin hormone, which has role in sleep and wake cycle. All of them, ADH, oxytocin, angiotensin 2, and also this melatonin. We'll talk about all of them in endocrinology, okay? So this was a little bit repetition about the this area which are called circumventricular organs, okay? So this was one function of the astrocyte, which it has role in blood brain barrier, okay? Now let me talk about another function of this astrocytes which has role in uh, glucose storage and glucose recycles. Okay, let me talk about another function of astrocyte which has role in glucose metabolism. Okay, let me talk about firstly about the glucose absorption from the blood to the central nervous system. This blood, this endothelial system. In endothelial cell, we have a transporter which is called GLUT1, glucose transporter type 1, and these transporters are insulin independent. They do not need insulin. So this glucose will enter to the endothelial cell, okay, through GLUT1. Then from the basal side, it also transported to the this area to the interstitium of the nervous system by GLUT1. Here is also GLUT1. Then this is astrocyte, this is neuron. Then this glucose is absorbed by the astrocytes also by GLUT1. So we have in both sides of endothelial cell GLUT1 and also in astrocytes GLUT1. Now this glucose goes to many ways. Okay, for example, firstly it goes out from the astrocytes by GLUT1. Here is also GLUT1, and when this glucose is absorbed by the neurons by GLUT3. This is so sensible to glucose. Okay, so this absorbed by GLUT3. This glucose then in neuron it converts through glycolysis to pyruvate, and then from pyruvate it goes through TCA cycle or Krebs cycles to produce a lot of ATP for neuron. That's one way. Another way for this glucose, it goes to storage in astrocytes in form of a glycogen, okay, by the process of glycogenesis. Okay, then now let me talk here. Okay, some of them are goes, for example, to pyruvate. They convert through glycolysis to pyruvate, and then through pyruvate they enter through TCA cycle or Krebs cycles and produces ATP, but less amount of glucose goes through this way. Okay, now let me tell you another thing. For example, this glutamatergic neuron, this a neuron which produces glutamate. When it produces glutamate, okay, so this glutamate then when it stimulates postsynaptic membrane, other of them are absorbed by these astrocytes. We will talk on this in next section. Okay, now this glutamate when enters to the astrocytes, it enters to it, it's a co-transporter to it sodium. Okay, this sodium then once again come out from the astrocytes by sodium potassium pump. Okay, and in this time these astrocytes sense that, oh, neurons need for energy because the functions of neurons are enhanced here now. So this, but because it needs a lot of energy for this sodium potassium pump, so now glycogen will be converted to pyruvate through the process of glycogenolysis, and then this pyruvate not converted to did not go to TCA cycle, but they converted to lactate. And this lactate is then transported from the or excreted from the astrocytes by a transporter which is called MCT1, monocarboxylate transporters type 1. And this lactate is taken up by these neurons by MC2 monocarboxylate type 2. Okay, now then this lactate will be converted once again, that I'll talk here about these enzymes. This lactate once again it will be converted once again to pyruvate, and then this pyruvate go to Krebs cycle and it produces a lot of ATP. So that's my target, that one important function of astrocyte is that it has in storage of glycogen. Okay, and when a neuron needs energy, so glycogen will be converted to pyruvate will be converted to lactate, and lactate then excreted and then absorbed by the neuron, and here it will be converted to pyruvate and then it will be converted to TCA or it go to TCA cycle or Krebs cycle and produce a lot of ATP. So this was the second important function of the astrocytes. Third important function of astrocytes is the easiest part of this one, that it has role in, for example, this is one neuron, this is one neuron, this is another neuron, okay, this one neuron, this another. When this neuron is stimulated, so during action potential that we will talk in future lecture, a lot of potassium comes out, okay, and this potassium comes out passively, okay. So when this potassium accumulates so much in extracellular area, so here we have astrocytes, and these potassiums will be taken by astrocytes, okay. These potassiums enters to astrocytes, and the level of potassium in extracellular area, it will come down. Then these potassiums, then they are transported by the gap junction from one astrocyte to another astrocyte, and after the termination of action potential, this potassium once again will come out. So that's why we say that another function of astrocyte is that it has role in potassium buffer system, okay, and also it has role in structural support of the neurons, okay, and also it has a role when the neuron tissue are go to damage, and they're damaged, and then the area where the neurons are damaged, they're filled by this astrocytes, okay. One another and last important function of this neuron is that it has role in glutamate and GABA recycle and in reuptake. Let me talk about this in full details. Okay, let me talk now about another function of astrocyte. It has role in GABA and glutamate recycle. Let me talk about this. It's so important. For example, this a neuron, and this neuron is glutamatergic. Okay, we have talked about this. So this neuron, firstly, it will release glutamate. This neuron, it will be release glutamate. Okay, this, for example, where is my blue marker? This glutamate, this is glutamate. When it releases glutamate, so this glutamate has EPSP action on this postsynaptic membrane, excitatory postsynaptic potential action on this postsynaptic membrane, and it will stimulate this postsynaptic neuron. Now, if this glutamate remain here for so many hours or for long time, so it causes excitotoxicity. It will stimulate this postsynaptic neuron so much, okay. So that's why here is astrocytes, and this astrocytes will take this glutamate inside to the astrocytes, okay, so faster, and this transporter here is called excitatory amino acid transporter, especially type 1, okay. So then this glutamate inside the astrocyte, it will be converted to glutamine. It will be converted to glutamine, okay. Glutamate had action of excitotoxicity, but glutamine can't work on this postsynaptic membrane, so it will convert it to glutamine by an enzyme which is called glutamine synthetase, okay, glutamine synthetase. Then this glutamine will be exported to the extracellular area by these glutamine transporters, especially these transporters, okay. Now this glutamine will be absorbed back by these glutamatergic neuron, but by its receptor, and then within this glutamatergic neuron, this glutamine once again it will be converted to glutamate, and here the enzyme is glutaminase, and there the enzyme was glutamine synthetase, okay. Now this glutamate go to storage and vesicles, it will be packed back and it will be reused. And another, this glutamine is going to the GABAergic neuron, okay. Now this glutamine will be converted to, once again it will be converted to glutamine by glutamine synthesis, okay, and then this glutamine will be converted by the help of this TCA cycle to GABA, gamma amino butyric acid, okay, and then this GABA is excreted and acts as a IPSP. For example, they will open chlorine channel, chlorine will come inside to the through this channel, and potassium will go outside to this and causes inhibition of this postsynaptic membrane. And like this glutamate, this GABA will also, it will be taken also by this astrocytes. In here, GABA will be converted by the help of TCA cycle or Krebs cycle once again to glutamate, and glutamate will be converted to glutamine, okay, and it will be excreted. So that was how that they had rule uh, in this circulation of GABA, or reuptake of GABA, and also reuptake of glutamate, okay. So this was an important function of this enzyme, okay. Here that this glutamate is converted to GABA. The enzyme here is glutamate decarboxylase, okay. So that was another function of these cells, okay. So now let me talk about another type of, this was so much about the astrocytes. So take it in your keep it in your mind that these metabolisms were not according to biochemistry. Here my target was that this is the functions of these astrocytes, because according to biochemistry they are so long pathways, okay, abundant in enzymes anyway. Now let me talk about another types of these cells or neuroglia, which are called ependymal cells, and then we'll talk about other cells. Let's talk about ependymal cells, okay. Keep it in your mind that astrocytes had rule in blood-brain barrier, and ependymal cells have rule in CSEP blood barrier. Let me talk about CSEP blood barrier. What is CSEP blood barrier, okay. Let me take this part, this fourth ventricle, okay. Let me take that here. This is fourth ventricle, this one is fourth ventricle, okay. And here I have, for example, blood vessels. So near the fourth ventricle I have blood vessels, okay. So barrier in fourth ventricle we have CSF, okay. So between this CSF and between this blood we have this barrier, and this barrier is called CSF blood barrier, okay. CSF blood barrier or CSF blood barrier, okay. Now let me talk about the structure of this thing. Firstly, from the blood side. In blood side we have endothelial cells. Here also we had endothelial cells, okay. But here we had tight junctions between endothelial cells, but here we don't have tight junctions between endothelial cell. That's one point. Another point, here also we have basement, basement membrane or basal lamina. Here we also had basement membrane. Third thing, here we had these pericytes and astrocytes. Here we have these ependymal cells. These are called ependymal cells. Here's the spelling, ependymal cells. It's called ependymal cells. And here between two ependymal cells, that's one ependymal cell, this another ependymal cells, between two ependymal cells we have tight junction here. So here we have tight junction between endothelial cell, and here we have a tight junction between ependymal cells. So here the blood CSF barrier is made of endothelial cell, basal lamina, ependymal cells, tight junction, okay. And in apical part of ependymal cells we have a cilia and cilias, okay. Okay, what are the basic functions of ependymal cells? One, it has rule in blood CSF barrier. It's one function of ependymal cell. Another function of ependymal cells, it has rule in production, that we will talk it in CSF production. In production of CSF, it will take it contains a lot of pumps, and it will take many things. For example, sodium, chlorine, water, all of them will come inside to this CSF, okay. And also it has role in circulation of C shape. It has role in circulation of C shape because it contain a lot of cilia. Okay, so that was the functions of ependymal cell, and keep it in your mind that another function of astrocytes are that they have rule in balance of water within the central nervous system because they contains aquaporin type 4. Okay, anyway, so these were the functions of the astro ependymal cells. Now let me talk about another function, another cell which is called microglia. Let me talk about this one. Microglia are the rest form of the macrophage. For example, when macrophage comes out from the blood, and then they go to store in some areas. For example, when they store in our bone, we call it osteoclast. When they are going to store in our liver, we call it Kaffer cell. When they are going to store in our alveoli, they are called alveolar macrophage. And when they are going to store in our central nervous system, they are called microglia. So microglia are here around the blood vessels and here in our central nervous system for phagocytosis of sub things. Okay, in some area of these ependymal cells, the ependymal cells are changed in their structures. Okay, for example, in area postrema, and here these changed ependymal cells are called they are not so tied, they can't they don't have rule in blood CSF barrier, and these are called tenocytes. Okay, let me talk about two important glial cells, oligodendrocytes and Schwann cell. Let me talk firstly about Schwann cell. Okay, for example, this is a neuron. Okay, this is an axon. When I turn around this axon, a plastic or something, and I cover it, so Schwann cell like this, they come around the axon and cover the axons, and this is called myelination. This is called myelination. The process of myelination is like this. In first step, in this step A, in first step, this axon is lying near this an axon, this a Schwann cell, this a nucleus. This axon is lying near the Schwann cell. In step number B, this axon dig into the Schwann cell. Okay, and step number C, this Schwann cell will make a layer around the axon, and this first layer is called mesa axon, and this first layer is called mesa axon. Okay, and then they will make many layers, and they will turn so much, and after this each layer is called myelin sheet. For example, this is an axon, this is an axon, this one turn, this one myelin sheet, this another myelin sheet, this another myelin sheet, and as a collectively this process is called myelination. Okay, and this outer form, this outer layer of myelin sheet, this outer, it's also called neurolema. Okay, neurolema, it's the outer layer of the Schwann cell. Okay, and even the inner layer is called mesa axon. These mesa axons are not compressed, their cytoplasms are not compressed like other layers. Okay, this is the process of myelination. Okay, now one Schwann cell is going to myelinate only one axon, or many Schwann cells come together and they are going to myelinate only one axon because they are so small. And another one Schwann cells are in our peripheral nervous system. Okay, they are not in our central nervous system. Another one, when we damage Schwann cell, it has a chance to recover itself. Okay, but oligodendrocytes are like Schwann cells, but oligodendrocytes are in central nervous system. One oligodendrocytes can myelinate many axons, 50 to 60 axons. Okay, and another one, if I damage oligodendrocytes, so it can't recover once again. Okay, so this was about the Schwann cell, this was about the oligodendrocytes, this was about the ependymal cells, and this was about the oligodendrocytes and astrocytes. And here is another cell which is called satellite cell. Okay, and it's not so important. Satellites acts like astrocytes, but astrocytes are in central nervous system and satellite cells are in peripheral nervous system. For example, this is dorsal. We have a ganglion around the spinal cord. It's called dorsal root ganglion. So they come and they cover this dorsal root ganglion, and it has role in protection, and also it has role for storage of energy for them. Okay. Great. So this was all about the neuron structure, and it was all about the oligodendrocytes and glial cells. In next video lecture, inshallah, we will talk about the resting membrane potential and action potential. Okay, so have a good time.